Okay, hello class. This is the start of one of my online lectures. This is the first chapter, and well, hopefully you've read the chapter. I want to point out that the subtitle of this chapter is Surprise and Awesome. Okay, so what they're saying is that the microbial world is often very surprising to us and can be quite awesome, and that includes bacteria being able to do things that no other kind of animal can do. Okay? So after you read this chapter, you should be able to describe at least 5 ways that microbes matter in the environment and to society, and there are definitely more than that, but how they impact us, how they impact the environment are very important. We can look at some of the major contributors of the beginning of microbiology and how they made their contributions. We can look at some of the contributions made during the golden age of microbiology. We can compare and contrast eukaryotic and prokaryotic cell structure and distinguish the major microbial groups. One of the things I want to point out at this point, there's a big difference between a eukaryote, something that has cell walls, and prokaryotes that don't have any internal membranes, and those are sort of two major ways of dividing life structures, but those are the two biggest ways of determining if they're different or not. And finally, we will look at some of the challenges that modern microbiology faces. Okay. One of the things that I think people sort of misappropriately know is that microbes cause disease, okay? And that sometimes is one of the major ways that people think about microbiology is only those things that cause disease. But microbiology is much, much more than that, and especially things that are just naturally present in the environment. Okay, here you see a picture with actual smallpox viruses, and then someone who has smallpox. Here are the bacteria that cause strep throat, Streptococcus pyogenes, and here's someone with strep throat down here. Actually, I think that the Streptococcus pyogenes is sort of a pretty bacteria. Okay, microbes are definitely very beneficial to the environment. Cyanobacteria are a type of bacteria that are able to generate oxygen, and the bacteria, in this case cyanobacteria, are able to generate all that O2 that we're going to need, that higher organisms need to make oxygen. Okay, of course plants make up the other 50%, but these cyanobacteria do make a good amount of them. Okay. We also have major decomposers that are used to recycle nutrients. It wouldn't really be possible to have landfills and things like that if we didn't have microbial action to break down the waste products. Okay, so why microbes matter? Okay, one of the things you might ask about is why the microbes matter. I mean, remember these are very, very, very small cells. Okay, if you wanted to, you could put 1,000 of those bacteria across a millimeter on a ruler. So how could something that small actually matter? Okay, now I'll give you sort of the punchline of that, and that is by sheer numbers. Okay, it's not just that they're not, they're very small. Okay, so they're not overpowering because of their size, but there's so many of them, and there can be so many of them because they are so small. Okay. So again, some examples of why the microbes matter on a farm, and one of the, or it's true of almost any higher organism, that you're going to have gut bacteria that are going to be aiding in digestion. The cows are a great example of this because, for example, the grass the cows eat can't be broken down by the person or the animal of the cow. They can only be broken down by the bacteria that are living inside the cow. Okay. We can also use microbes as natural pesticides, and that is just a lot safer for the environment to be able to use those microbes. Okay. In our food production, there are basically two major ways that bacteria are affecting food. One is that the food is being produced, so something like yogurt or sauerkraut, that the bacteria used to make the to make the food product. The second part would be food degradation. You know, if you leave something out too long, that the bacteria are going to take over there and ruin the food. Okay. There are a couple good examples of food fermentation, sauerkraut or beer, and I tend to think that most of you at least drink some alcoholic product, and you can't have alcohol without microbiology. The bacteria are important in bioremediation. We can use one microbe to clean up the waste of some other system. I've done a lot of work in bioremediation, actually bioremediating a acid mine drainage site where the water is very low in pH, very high in iron, and we can use the bacteria to clean up the system. Okay. In any modern city, we have sewage treatment plants that we wouldn't actually be able to survive without the breakdown of all that waste. Okay, you know, I would guess that most of you don't really think about what you flush down the toilet and where that goes and how it's gotten rid of. Every city has some sort of way of treating that sewage so that it can be gotten rid of. Okay. Now we can use the bacteria to make some pharmaceuticals. There you see a fermenter, and that sort of yellowish liquid is a solution containing all the bacteria in it. And one of the very first places that bacteria were used were in the production of insulin. Okay, and since about 1983 or so, I have been using insulin that is made by bacteria. And without that insulin, I wouldn't be able to survive. So it's actually a really good system because it's one of the best systems to be able to make some peptide or protein hormones like that. Okay, another protein hormone that is used pretty widely is growth hormone. Okay, we can use bacteria to make vaccines, and they're just a few examples. Now, on the human body, okay, you are the minority player on the human body. The number of cells in a human is way less than the number of bacteria that are actually in you. Okay, and over the course of the semester we'll go over all these examples, but there's something called the microbiome, okay, and that's where each area of the body can have certain bacteria that are constantly living there, okay, and I think we all sort of know the ones that are listed there, the oral, the skin, as being major places for bacteria to live. The skin is a major player in just being a place where bacteria can live. Okay, so obviously any field, would not be a major player if there weren't people who pushed the fields forward. So here are some of the major pioneers of microbiology who are important to remember. Okay, one of the first people to look at bacteria under a microscope was Robert Hooke. Okay, there was someone about 100 years before this who had seen the bacteria first, but Hooke was really the first one to really observe them. Okay, and so he was able to draw some things through his microscope. One of the most famous slides he has ever done is of cork, showing all the different cavities that are in a cork. Okay. He also was able to observe molds. It might not have been appreciated that molds were a type of microbe at first, but Robert Hooke was certainly able to show that. Okay. Leeuwenhoek was the first person to ever observe bacteria, and it's interesting, at the time microbiology was mostly a hobby. Okay, so by profession, Anthony van Leeuwenhoek was a clothes merchant, and he actually made his own lenses and made his own simple microscopes. These were very, very simple devices, just a simple one lens system, so it basically sort of acted like a magnifying glass, but he was able to see quite precisely. Okay. Even though he was not British, he would send his observations to the Royal Society in England to try to get validation for his discovery of the bacteria. Okay. Leeuwenhoek called these things the animalcules because to him they look like little tiny animals, even though, cause they were moving like they were animals, but they were very, very small. Okay. Now, the first person to really does do a study of microbiology was Louis Pasteur. Okay, and pasteurization obviously is named after him. He's the one who discovered that, and Pasteur came up with something called the germ theory of disease, and he was actually studying microbiology because he wanted to find out why wine's sour. Okay, pasteurization. And one of the things that Pasteur is sort of noted for is that he refuted this theory of spontaneous generation. So at the time, people all over science, what they really cared about, or they always thought that all life came from spontaneous sources. Okay, if you got the right conditions, you would get these life forms, and they really had no idea about things having parents. Okay. Um, okay, so I told you that Pasteur was able to disprove spontaneous mutation, or sorry, uh, spontaneous generation, and so he did two basic experiments. Okay, he would take a flask and put some media in it, so something the bacteria like to grow in, sort of like a soup, and he would boil that solution, and we already knew that boiling water would kill all the organisms inside there. Okay, so in the first thing, he would take a flask and leave it open to the air, and the second one, he would seal the flask. Okay, and then both those bottles were allowed to sit, and we would see what would grow in them. Okay, so after a certain amount of time, the one exposed to the air would grow lots and lots of bacteria, whereas the second one that was sealed never actually grew any organisms in it. And one of the things I think is really interesting, at the Pasteur Institute in Paris, they will say that the jars that they have there now are the original jars that Pasteur set up, and they still don't have any contamination in them. Okay, so they're saying 200 plus years that this experiment is still working, that you don't get any spontaneous generation. When you think about the Koch postulates, okay, again, you have to isolate the organism and be able to cause that disease again in a new animal. Okay, now there's a real problem with that. Okay, and this is one of the things why there are people who will say that, say, HIV doesn't cause AIDS, because you can't do that experiment. Okay, I mean, there are a couple of reasons why you can't do that experiment. One is that you really can't isolate HIV out of a whole organism, and you would also have to infect another human with whatever you got. And again, that's just not ethical to give someone a fatal disease. Okay, so you know, there's people who will say that the Koch postulates are the ultimate way to prove if an organism causes a disease. But like I said, there are definitely problems with that, because it's just not ethical to cause a disease, especially a lethal disease. Okay, we just went through a few examples of some of the pioneers in microbiology. I'm now going to talk about some of the people who are important in the golden age of microbiology. So the golden age is really defined as that area where microbiology became a major field within itself. Okay, so this is from about 1850 to 1910, and they include Pasteur for being able to talk about cholera and also develop a human rabies vaccine. Koch for finding out that tuberculosis causes tuberculosis. And he was also important in developing a diphtheria vaccine. Ross showed that mosquitoes transferred malaria. Bruce that TT5s transmitted sleeping sickness. Ogata that bubonic plague can be transferred from rat fleas to other rats or to humans. And Rocky Mountain spotted fever can be transmitted by the bacteria Rickettsia, and that bacteria is named after the discoverer, who is Ricketts. Okay. Reed looked at mosquitoes transferring yellow fever. Winogradsky was a Russian who did a lot of environmental microbiology, and he was able to show that carbon dioxide can be used to make sugars from plant sources. Okay. Bendrick used bacteria to make nitrogen for plants and show that the nitrogen from plants are from a certain class of bacteria called nitrogen fixing bacteria. Okay. So in the 21st century, we use a lot of antibiotics to cure infectious disease. And this was a major discovery, and obviously saved countless lives. But now in the 21st century, we're having this problem with antibiotic resistance. Okay, the bacteria are always able to mutate to give antibiotic resistance. Okay. Also in the 20th century, we were able to use an electron microscope, and an electron microscope are really the only way to be able to see viruses. Okay. Now, let me actually put that. Okay. Like I said earlier in the presentation, there are two major classes of microbes. We can either have eukaryotic cells or prokaryotic cells. Okay, and in a eukaryotic cell, you have a nucleus, okay, that contains all your DNA, and then you have the cell wall all the way around, and even internal membranes inside of that. Okay, these cells are quite large. You can actually see them without a microscope, whereas in bacterial cell, okay, a prokaryote, these bacteria have the DNA, but it's not in a nucleus. Okay, that's actually what prokaryote means, without a nucleus. These bacteria have their DNA without a nucleus, and these are quite a bit smaller. Like I said, you can fit about 1,000 of these across the millimeter in size. We can also have viruses, which are just the protein coats and the nucleic acid. Okay. There are a large number of microbes. We can have bacteria, we can have viruses, fungi, algae, um, protozoans, all those things all count as microbes. One of the sort of easiest definitions for what a microbe is that is anything that you have to see under a microscope. Okay, definitely individual cells of a mold or a bacteria or something like that, you have to use a microscope to be able to see them, so they truly are microbes. Okay, so you might think that in modern everyday life that we've basically solved most of the problems that we have. However, this just isn't true. So we have some remaining challenges. We're going to have disease outbreaks, okay? And here's a map from the CDC showing how different outbreaks occur. On this map, the darker the color, the more cases are in that state. And there is a national survey in this country that all reported cases of certain diseases have to be reported to the government. Okay, so we can sort of make accurate maps like this. And this information is required of doctors every week. Okay, they have to notify if they've seen anyone in their office with one of these diseases. Okay. I sort of mentioned it a little bit before, but we do have a problem is that bacteria are becoming more and more resistant to antibiotics. So while at the turn of the 20th century, antibiotics became all the rage and were so important in wiping out diseases because they were killing the bacteria, now we're getting all these antibiotic resistant bacteria that you just can't kill anymore. Now we can monitor diseases worldwide, and it's sort of hard to imagine anymore that a disease will stay isolated to an area. I mean, there's so much plane travel and stuff like that. You're going to get outbreaks basically worldwide. Okay. So some of these things that you should have learned from this is, well, actually the second point I like among the best is that there are no stupid bacteria. Okay, the bacteria will do anything to survive. Okay, and the better we are good at letting those bacteria do what they want to do, we will find more and more ways to be able to use them. Okay, and like I said, we have lots and lots of bacteria in and on our bodies, and so if you take care of them, they will take care of you.